So we just got finished talking about PI3 kinase and how it is regulated, and now let's talk about how it is dysregulated often in human cancer cells. So just to briefly summarize, right, PI3 kinase is regulated by receptor tyrosine kinases or RAS-GTP, which can allow PI3 kinase to become active and phosphorylate PIP2, converted to PIP3. We covered that in the previous video. In many human cancers, PI3 kinase is overactive. If PI3 kinase is overactive, we'll talk about how, but let's talk about what's the result of that. If PI3 kinase is overactive in human cancers, then it is constantly phosphorylating PIP2, converting it into PIP3. And when PIP3 is present, high levels of PIP3 signals to lots of things, including the AKT pathway, which will allow the cell to keep going through the cell cycle. It will inhibit apoptosis. It will allow proteins to produce high amounts of protein to replicate. It affects the metabolism of the cell. So if PI3 kinase is overactive and producing a lot of PIP3, then cells are going to be in this pro-growth, anti-death state. And that's bad, right? If we want to treat cancer, we got to stop PI3 kinase. So first, let's talk about how PI3 kinase can be overactive in human cancers. And really, there are two ways to think about this. Either mutations can occur in the genes that code for PI3 kinase, or mutations can happen in genes that code for proteins that regulate PI3 kinase, or what we would say is genes that are upstream of PI3 kinase. So let's start with that second one first, about mutations in genes that code for proteins that are upstream of PI3 kinase. And we just saw in the previous video, receptor tyrosine kinases regulate PI3 kinase. So if there are mutations in receptor tyrosine kinases, so we talked about that in a previous video. You could have amplifications of genes that code for receptor tyrosine kinases, point mutations that are activating the kinase activity of receptor tyrosine kinases, autocrine or paracrine overproduction of growth factors. Either way, in many human cancers, receptor tyrosine kinases are overactive and phosphorylated constantly on their tyrosines in the cytoplasmic tail. That would lead to PI3 kinase binding directly or indirectly to those phosphorylated tyrosines and constantly converting PIP2 to PIP3. So mutations in the receptor tyrosine kinases, or things that regulate them, could lead to overactivation of PI3 kinase. We said that RAS regulates PI3 kinase. When RAS is in its GTP-bound form, it will bind PI3 kinase and allow PI3 kinase to come up to the plasma membrane and convert PIP2 to PIP3. Remember, RAS can be mutated in human cancers. You could have point mutations in RAS, which abolish its GTPase activity. And if its GTPase activity is abolished, it can never remove that terminal phosphate off the GTP and go back into its GDP-bound mode. So if RAS is mutated, then and it's stuck in this pro-growth version where it's always bound to GTP, that could be activating PI3 kinase and causing PI3 kinase to constantly phosphorylate PIP2 and convert it into PIP3. So those are mutations in genes that are upstream of PI3 kinase. You could also have mutations in PI3 kinase. Let's see what that looks like. So recall from the previous video, the genes that code for PI3 kinase. There is the catalytic subunit P110, which is coded for gene PI3K for catalytic subunit, A for alpha, but we're not going to get into that. And then the regulatory subunit PI3K for regulatory subunit, number 1. So you get mutations in these genes. So what kind of mutations? Well, we'll just say right now that PI3 kinase is an oncogene. So oncogenes are typically mutated, and when they're mutated, they are constantly active. So these would be activating mutations. So what kind of mutations are we talking about? Two major types of mutations. One is gene amplification. So when a gene is amplified, then cells end up typically producing abnormally high levels of protein. We call that overexpression. So the PI3K3CA gene is often amplified in human cancers, which will lead to overexpression of the protein, and we'll see what that does in the next slide. Another very common mutation in human cancers, not all cancers, but some human cancers, a point mutation in the gene that codes for the P110 subunit. Now, again, what do mutations do to proteins? It depends. A point mutation can abolish the enzyme activity of a protein. They can also supercharge or activate the enzyme activity of that protein, depending on where the point mutation is, depending on the effect of the three-dimensional structure of the protein. So mutations don't always abolish the function of an enzyme. Mutations can supercharge the function of that enzyme. And that's exactly what happens here. Point mutations in the catalytic subunit of PI3 kinase actually activate its kinase activity. So they're called gain of function mutations. So these are the two common mutations that you find in human cancers, either an amplification or a point mutation. Either way, we're going to produce too much active PI3 kinase. And let's see what that's going to look like in the next slide. So again, in a cancer cell, we talked about mutations of receptor tyrosine kinases and mutations in RAS. Now let's talk about mutations in PI3 kinase. So let's say that the catalytic subunit is amplified. So that would lead to the abnormally high production of the protein P110, the catalytic subunit. That would be bad to have too many of these into the cell, because if you had too many of these, at some low rate, they will encounter PIP2 and convert it to PIP3. They're not supposed to. There's supposed to be a low level of this protein, but there's an abnormally high level of this protein. So at some rate, it will be converting PIP2 to PIP3, even though it's got no signal, even though it might be missing a regulatory subunit. Having too much of this protein is not good. The other common mutation, again, is that point mutation that makes a P110 subunit that is highly active. So again, cells might not be receiving signals from receptor tyrosine kinases or RAS, but these mutant P110 catalytic subunits will convert PIP2 into PIP3, even though there's no signal coming into the cell, even though this is not an ideal situation. But having abnormally high levels of the P110 subunit or having a gain of function point mutation in the P110 subunit can cause enough PIP2 to be converted into PIP3 to get the cell to go into the cell cycle, to put the cells in an anti-apoptotic state and allow cancer cells to grow and survive. So this is that mutations in PI3 kinase can lead to cancer. What about targeting this pathway for treating cancer? And that's exactly what happens, right? So there are drugs that can treat cancer that target PI3 kinase. So PI3 kinase, it is a kinase, and in cancer cells, it could be overactive. So what could we use to inhibit a kinase? The kinase inhibitor, right? So there are biochemists who have designed kinase inhibitors that bind the P110 catalytic subunit, the ATP binding pocket, like many kinase inhibitors do. And if these kinase inhibitors are present in the cell, what are they going to do? They're going to bind to the 110 subunit. They're going to prevent the 110 subunit from phosphorylating PIP2 and converting into PIP3. So now, instead of having high levels of PIP3, we have low levels of PIP3. So now the cell will stop getting that signal to go through the cell cycle and can actually trigger apoptosis. Because again, remember, PI3 kinase feeds into, PIP3 feeds into AKT pathway, and we'll learn AKT regulates apoptosis as well as cell survival, cellular metabolism. So there are a whole class of drugs that are PI3 kinase inhibitors that will regulate the levels of PIP2 and PIP3 by inhibiting PI3 kinase. And many of these are approved for treating human cancers. And I'm just going to write out a couple of the names. These are the generic names of PI3 kinase inhibitors. I'm not going to try to pronounce them. I'm terrible at pronouncing these names. That just seems like a mix of vowels and consonants. But I do want to point out something, for those of you who are interested in pharmacology, is the suffixes for drug names commonly refers to the mechanism of action of those compounds, or the targets of those compounds. And you'll notice all of these drugs end in LISIB. So LISIB, that suffix refers specifically to compounds that inhibit PI3 kinase. And again, PI3 kinase is a very different kinase than kinases we've talked about before. We've talked about tyrosine kinases, we've talked about serine threonine kinases. Those are protein kinases. PI3 kinase is not a protein kinase. It is not phosphorylating a protein. It is phosphorylating a sugar or a lipid, depending on how you want to look at PIP2. So PI3 kinase is it's a different kind of kinase. And if you look at the homology of PI3 kinases and protein kinases, they're actually quite distant in their homology. But suffice it to say, PI3 kinase is a real target for drugs used to treat cancer. And again, hopefully this video will help you understand why you would want to inhibit PI3 kinase in cancer cells.